a center of excellence, a nurturing space for innovation, creativity, and academic freedom. This is the University of the Philippines, Los Baños. As a constituent university of the University of the Philippines system, UPLB is a leading national higher education and research institution in various niche areas. Grounding itself on the needs of national development, UPLB cultivates well-rounded and critical leaders who are ready to lead breakthroughs and innovations. Through its industrial and academic partnerships, UPLB propagates its gains to advance inclusive development in various sectors. An educational institution that upholds honor, excellence, and public service.
A center of knowledge, innovation, history, culture, and biodiversity. This is the University of the Philippines, Los Baños. UPLB found its home at the foot of the legendary Mount Makiling in Los Baños, Laguna, a short distance from Laguna de Bay, the country's largest lake. It has its beginnings in 1909 as the UP College of Agriculture, a tent school for a handful of students. A year after, the Forest School, or what was to become the College of Forestry, also set up its home in Los Baños. In 1972, the UP College of Agriculture and the College of Forestry, that had by then become renowned institutions, were granted full autonomy as the University of the Philippines, Los Baños. Instruction, research, and extension in agriculture and forestry in the country took root and flourished in UPLB. Through its strength in these two fields, UPLB established niches in various areas: biotechnology, engineering, veterinary medicine, natural resources conservation, environmental science and management, human ecology, food science, economics and management, public affairs, and development communication. It drew its strength in the arts and other fields of science, from harnessing its expertise and human resource in the biological, physical, and social sciences. Today, UPLB degree programs continue to dominate over its counterparts. Over the years, UPLB has developed graduate programs that continue to attract students far and wide, including collaborative programs with international universities in food security, biodiversity conservation, and development. To make graduate programs more accessible to professionals, UPLB offers them off-campus. Recently, UPLB established the UP Professional School for Agriculture and the Environment in Mindanao, the country's food basket, in order to help build the knowledge capital that the country needs to attain food security. Instruction in UPLB is enhanced by a strong and vigorous engagement in research and public service that enables it to produce knowledge and generate technologies and innovations that promote sustainable living, making it a strong force in the war against hunger and poverty in the country. Extension and public service are the mechanisms by which UPLB brings this knowledge and technologies to the people, as well as for it to know what they need and to help shape policies in the country. UPLB's reputation as a graduate and research university was formed through the continuous aspiration for excellence and the mutual reinforcement between instruction, research, and public service and extension. It brings in partnerships with local and international universities and provides mobility to faculty members, researchers and scientists, and students and enhances their competitiveness. Instruction, research, and extension and public service in UPLB reflect the dynamism, honor, and excellence that its forebears set as a standard for its leaders to aspire for.
The world is in a constant state of change now more than ever. It's a world that requires resilience, flexibility, and innovation to overcome challenges and to pursue growth. That is why the University of the Philippines Los Baños is pursuing future-proofing initiatives to provide agile leadership and develop local and global communities amidst shocks and disruptions in an ever-changing world. Guided by compassion, an openness to working together, and the daring to seek out breakthroughs, UPLB constantly pushes for academic excellence, produces relevant research, establishes academic and industry partnerships, and cultivates creativity, critical thinking, and innovation. To keep up with the fast-changing world and to strive for inclusivity in development, UPLB seeks to level the playing field and expand avenues for acquiring new knowledge. It is reshaping the learning experience, enabling students and educators to continue with the learning process in vastly different learning conditions. It is expanding its academic programs and supporting initiatives to create a conducive learning environment. To address the growing complexities of modern problems, UPLB continues to break barriers across disciplines to produce cutting-edge innovations and solutions that serve the needs of various communities. It is taking on challenges in food security and sustainability and coordinating research efforts to direct the university's resources toward critical research areas, making sure that the university-generated knowledge can reach its intended stakeholders and be applied to real-life contexts. UPLB is dedicated to its commitment to contribute to public good and social welfare as it continues to work with the local government and the community to address their most pressing needs. Amidst its engagements, UPLB remains as one of the top regional and global universities and continues to be recognized in the country and across the world. As the national university, UPLB aims to be a steadfast partner in development, as a producer of new approaches that minimize the cost and impacts of future shocks, and by honing leaders committed to knowledge creation, innovation and cutting-edge research, and public service. This is UPLB's roadmap to be an impactful institution in a constantly changing landscape. Responsive, innovative, future-proof.
Okay. Right. So good morning po sa ating lahat. Blessed Wednesday morning. So today is December 7, 2022, and Team LRC is back for a new set of learning sessions entitled GENE-ius ka talaga, a tutorial webinar series on genetics. So kumusta po ang bawat isa sa atin ngayong umagang ito? So sa ating mga nasa Zoom na napakaaga, kumusta naman po kayo? Nag-breakfast na ba kayo? Si Jay Luis Infante, to napakaaga natin na participant. Very eager ng matuto sa ating session ngayong umagang ito. Jay, kumusta ka ba ngayon? Ayan. Siya ay very ready na matuto sa ating lessons ngayon. How about the others? Kumusta po kayo? It's a very nice Wednesday morning. No? Dito sa Los Baños ay napaka-araw. Nawa ay patuloy siya magiging maaraw at mahirap magpatuyo ngayon. At kapag maulan-ulan ay yes, tambak ang ating mga labada. So anyway, so tayo po ay sasabak muli sa isang talakayan tungkol sa genetics. So as I've mentioned yesterday, we tried to compress lahat ng mga vital na topics on genetics in 3 sessions. So talaga namang kahapon, ang ating session ay very fruitful dahil we were able to discuss about the different chromosomal principles and the basics of heredity. At tayo ay nakapaggawa ng mga problem-solving activities kahapon. And si Sir Eman ay talaga namang natuwa sa high engagement ng ating mga participants. And there will be more of that today. Okay? Kaya naman sa mga nakatutok sa atin sa FB Live, magandang umaga po sa inyong lahat. Let us know kung saan po kayo nanonood ngayon. So, good morning, Ma'am Emeline Espiritu. Hi, Tita Emmy. Yan, nandito rin po si Oslek Evangelista, John Lloyd Subtinente. So, hello, good morning po. Catherine Villoan Garcia, good morning. Yes. Sir JV, yes, it's still the same, po. Antal dito? Same Zoom link po. Yes. So you can join us po via Zoom. Okay? So, all right. Kung wala pa po dito ang inyong mga classmates, students, and colleagues, itag na po natin sila sa FB Live or i-share niyo po ang ating Zoom link para po sabay-sabay tayong matuto sa umagang ito. As of this morning, we already have around 312 registered participants, very active participants who are very willing to learn more or to refresh their knowledge on genetics. So from the social demographics, we saw that plenty of students across different constituent universities of the UP system, as well as other colleges and high schools across the Philippines are joining us in this tutorial webinar series. So I hope on board na po tayong lahat. It's 9:05 in the morning. Welcome to GENE-ius ka talaga, a tutorial webinar series on genetics. So genetics is a subject that both interests and challenges students and teachers. So which is why for this tutorial webinar series, ang Team LRC po ay nag-prepare ng 3 session para i-discuss and for us to fully appreciate the different fundamental lessons in genetics. And for our students here in UP Los Baños, sa mga nagbabayo 30, ito ay parang review na rin natin sa mga susunod nating mga long exams, no? For Bio 30. And as part of the center's team learning sessions, we provide these tutorial sessions to serve as a review and prepare our students for their upcoming assessments. Okay? So I am Joshua Michael Jonas, University Research Associate and Tutorials Coordinator of the UPLB Learning Resource Center, and yours truly will be our session's moderator. We are now on our second session entitled You're My Genotype, How Genes Function Within Us. So sino nga ba yung mga type natin? Pero for today, we will be discussing about genotype, and more specifically, genes in general and their functions. So we'll be learning about the different aspects of our genes, their specific functions and characteristics, as well as the molecular basis of inheritance. So just a few friendly reminders for our session, for a smooth flow of our session number 1. Please prepare your pen and paper and take down notes and actively participate in our discussion. Second, keep your microphones on mute and cameras off unless instructed to avoid distracting the speaker and your fellow participants. You will be given a chance later during our photo op. Use the Zoom chat box properly and responsibly. At all times, please keep your comments helpful and considerate to the speaker, moderator, and to your fellow participants. 4, if you have questions, feel free to write them down below our comments section or chat box and make sure they are relevant to the topic this morning. Sir Eman will be gladly to answer or address these questions. And lastly, please do not forget to answer the evaluation form. After the session, our team will deeply appreciate your comments and suggestions. Please also be reminded that the deadline of responses will be until 10 p.m. tonight. For those who will not be able to join us today because you have equally important matters to attend to, perhaps you have a meeting or another class to attend, you can watch the replay of our sessions on our Facebook page. So we now have 106,000 strong followers. So we are truly grateful po sa Team LRC. Likewise, you can also watch the recorded sessions on our YouTube channel. Just search for UPLB Learning Resource Center on YouTube. And please continue to like, share, and subscribe. Okay, and we also invite you to connect and follow us on Twitter, TikTok, and LinkedIn. Kindly use our official hashtag for this tutorial webinar series, which is hashtag GENE-ius katalaga, para mabilis po nating makita ang lahat ng videos ng ating series using that hashtag, together with hashtag Team LRC. Okay, moving forward, let's have a short energizer. 9 pa lang naman na umaga, so tayo ay mag-unat-unat muna ng konti ng ating brain cells. Na? So if you have already attended our previous sessions, you know the drill and you know what to do. Sa mga ngayon pa lamang po makakasubok or makakatry nito, madali lamang po. First, open a new browser or a new window and go to www.menti.com. And second, type in the key code that you see on your screen, which is 73016385, and answer the questions that I will be flashing on the screen. Okay? So allow me to share my screen for our energizer. Okay? Ayan. Okay. So, how are you feeling this Christmas season? So, tayo ay malapit natin makalahati ang Disyembre, at ano ba ang inyong nararamdaman as of this moment, December 7? Now, are you feeling happy, excited, sad, stressed, or meh, lalang? Ano ang ating nararamdaman dito sa ating mga nasa Zoom and Facebook Live? Ayan. Let's see. Join pa tayo sa ating Menti. Natin makasa 10 years pa. Ilan ba tayo dito sa Zoom? Sa 22 tayo sa Zoom. Okay. Happy, excited, stress? Ano bang nararamdaman ng inyong mga puso at diwa ngayon darating na Pasko ng 2022? Ayo ba ay ready na sa inyong mga Christmas party? Take note, sabi ng DOH ay tayo ay mag-iingat pa rin dahil nandyan pa rin ang COVID. Sa ating mga guro at mga manggagawa, nakamit na ba po natin ang ating inaasam na text message ng ating cashier ngayong Disyembre? Yun ang isa sa mga nakakapagpa-excite sa atin, no? Ano pa ba? At least wala na mag-sad or may okay lang yan. Stress is meron tayong good and bad stress. So hopefully good stress itong tatlong nagsagot sa ating Mentimeter, no? Okay. So at least mas mataas ang excited kaysa sa stress. So excited and happy ang ating mataas. Hopefully this is a good feeling that we will be experiencing and exhibiting throughout December before we end this year of 2020. Okay. Second question, ito. Medyo review natin. What were your takeaways from yesterday's session on heredity and chromosomal principles? Ang daming na-discuss kahapon ni Sir Eman. Ano ba yung mga kumbaga, ina-sticky note natin ng mga points kahapon. No. Sige nga. Sina Jay, ano ba yun yung mga na-recall kahapon, Jay. Sino ba din? Magtatawag ako. Pero ilagay nyo lang dyan. Sa testamenting method. Si Clark, ano bang mga nakuha nyo? Tegi method. Feeling ko isa to sa mga tumatak Sir Eman the Tegi method, no? Na ang ibig sabihin ay, let me see kung na-recall ko, treat every, ano yung G, Sir Eman? Oh my God.
Take each gene
Each
pair.
gene pair individually. Ayun, Tegi method. Ayun. Si,
Wasn't able to check. Ay, sorry.
oh, Rhea, ano kaya? Sherlyn, ano ba yung mga na-recall ninyo kahapon? Segregation, no? And assessment. Pati yung ano, Sir Eman, na hindi palagi ang solusyon ang Punnett square. No? Sa ating mga problem solving. Sometimes it's better to use the long method. Manx cat. Oh, yung Manx cat, no? Ngayon, tapon ko lang din nalaman dyan. Over-dominance and co-dominance. Yan. Okay? Sige. So talagang ano, malaki dito sa ating word cloud yung Tegi method. So yun talagang natanda. Baka ngayon, Sir Eman, baka may iba kayo mag-share na acronyms or mnemonics, no? Okay. So yun talaga yung mga nang tatatak sa puso at isipan ng ating mga mag-aaral. Okay? So, hindi ko na papapatagalin pa. Let's now go to the main part of our session. So, our speaker for this morning is an assistant professor at the Genetics and Molecular Biology Division of the Institute of Biological Sciences, College of Arts and Sciences here in UPLB. So, here to share with us how genes function within and the molecular basis of inheritance. Ladies and gentlemen, dear friends, let's welcome in the Zoom space Assistant Professor Emmanuel T. Galang. Sir Eman, good morning po.
Morning. It's really nice to see familiar faces and at the same time new faces in our Zoom space. Sir Josh, maraming salamat po for that very warm introduction. I'll take it away.
Thank you, sir.
It's really nice to see colleagues as well in the field of education really taking the time and investing the efforts to attend to these kinds of webinars kasi this is all for the sake and the benefit of our students. So maraming salamat din po and para po ito lahat sa bayan. So magandang umaga sa ating lahat. Before I proceed, 30 seconds, set up lang ako ng screen because I'm going to do chalk talk again just like how I did it yesterday. One sec. So for today, we will be focusing on the genetic material. We are all well aware kung ano yung nangyayari sa ating genetic material, no? During chromosomal behavior and movement at mitosis and meiosis as discussed from last meeting. So if you recall, I'm sure that you will recall. So alam natin na whatever that is passed on from one generation to the next, lahat ng genetic information na yon are all confined in the chromosomes as we discussed yesterday. And from our discussions, we have found out that your chromosomes are, balik tayo sa slides ko yesterday, or sa scribbles ko from yesterday. We all know that, 19 pages pala tayo kahapon. There. We all know that your chromosomes contain very particular parts that make them distinguishable to the naked eye upon first sight. So makikita natin that these contain specific parts and the genes are confined within the arms of the chromosomes. All right. And on top of that, a chromosome number is the one that tells us kung gaano kadami yung chromosomes sa isang organism. So generally, a chromosome number is a two-sided equation that involves the left side of the equation that tells you as to the number of sources of your genetic material. So since we are 2n equals 46 as individuals, kasi 2n equals 46 in humans, this tells us that 23 of our chromosomes come from one parent, while the other parent will constitute the other half. We have also found out that your chromosomes are indeed dynamic. So these images here show the fact that these are all pertaining to the same chromosome across different stages in the cell cycle. So makita natin na as much as the cell changes through its maturity or through its maturation process, we also come to realize that with these changes come the changes in the physiology of your chromosomes. Makikita natin na may time na ninipis siya, may time na kakapal siya, may time na dodoble siya, and may time na maghahati din siya at the same time. All right. So we have discussed different types of cell division, no? First, primarily involving body cells, and this type of mutation is referred to as mitosis. We have elucidated on the fact that mitosis itself is responsible for equational division. Ibig sabihin yan is that the chromosome number found from the original will be maintained in your daughter cells. So therefore, if we started with 2n equals 4, eventually all of its daughter cells will lead to 2n equals 4 because division is equational in mitosis. Ano sa tingin niyo yung importance ng equational division sa mitosis? Anybody in the chat box? Ano sa tingin niyo yung importance? All right, sige. I'll help out. Yung importance ng equational division sa mitosis is that it is the one responsible for maintaining the chromosome number across all cell types in the human body. Remember, we all started as single-celled individuals after fertilization. So once the sperm, which is a haploid with n equals to 23 na chromosome number, meets with an egg cell with a chromosome number of n equals to 23, you put them together. Upon fertilization, we restore the regular 2n equals 46 chromosome number that we have as individuals. So the 2n equals 46 na chromosome number, upon fertilization, is maintained up to your state right now after billions of cell divisions. And it will still maintain the chromosome number 2n equals to 46 regardless of what cell type you see in your body except for sex cells. And the main reason for that is mitosis. So mitosis is the one responsible for maintaining a homogeneous chromosome number across different cell types in your body, except for your sex cells, of course. So meiosis is a little bit different. Because with meiosis, nakita natin na after 2 rounds of cell division, from a diploid state, say for example, as humans, we have 2n equals 46 na chromosome number. After the completion of the 2 rounds of meiotic division, you will end up with a haploid, n equals 223. All right, so this is one of the major differences between meiosis and mitosis. The reason for that is because the type of division occurring in meiosis, specifically at anaphase 1, is referred to as reductional division. It is at this point wherein wherein the chromosome number will be halved in the process. Okay, so makikita din natin dito that eventually you will produce 4 daughter cells from 1 cell of origin in meiosis. So makikita natin sa ating mga images discussed from our previous discussion that mitosis is actively occurring in your cells, specifically during times of growth, during times of repair, and at the same time in common cells that are renewed and replaced time and time again. Classic example niyan is that, are you guys familiar with chemotherapy? When a patient undergoes chemotherapy, it has the tendency to kill off cells. And the cells that it generally targets are those that are rapidly dividing. Cancerous cells man yan or non-cancerous cells. Kaya kapag yung person is undergoing chemotherapy, one of the main characteristics of a person undergoing that kind of treatment is the lack of taste. Mawawalan sila ng panlasa. Because there is active cell division in the cells of the linings of our mouth. But if a person is undergoing chemotherapy, iha-hamper niya yung rate ng cell division such that maapektuhan yung kanyang panlasa. So remember, chemotherapy attacks rapidly dividing cells, which does not only include cancerous cells but regular cells in the body. Ayan. So dito makikita din natin the process of meiosis occurring in 2 rounds. So the first succession, makikita natin it produces 2 daughter cells. And then eventually the 2 daughter cells will undergo another round of cell division, thereby producing 4 daughter cells in the process. Okay. So because of this meiotic behavior and meiotic behavior, this supports Mendelian principles of heredity, such as segregation and independent assortment, just by looking at how the chromosomes behave and separate during the process of meiosis. So the first Mendelian principle discussed yesterday was the principle on segregation, wherein alleles of a gene pair will definitely separate in the process. All right. So say, for example, if an individual is a heterozygote, its gametes produced will only carry either the dominant allele or the recessive allele, but never both. Okay? So next, we also have discussed how this affects independent assortment because chromosomes from genes from different homologous pairs or different bivalents can assort independently from each other. So nakita natin sa illustration na to that the chromosome carrying the small b allele can assort with the chromosome carrying the small a allele or a chromosome carrying the big A na allele. So it happens independently. So therefore, there are a lot of possible combinations occurring across multiple types of chromosomes in the process of meiosis, thereby adding to variation as we see it. Okay, so sample problems natin na-solve, no? And always remember, whenever you're faced with these kinds of questions, never be tempted to automatically go to Punnett square right away. Usually, the best technique here, yung sinagot nyo kanina sa Mentimeter, is to take each gene pair independently so that you will just multiply the probabilities from these independent events which will lead you to what is being asked. Ayan. So eto nakita natin kahit 3 na gene pairs pa yan, kayang-kaya yan ng taking each gene pair independently. Ayan. So dihybrid cross, di na natin ito napakita. No? And also, there was this question asked yesterday kung paano na-form yung bar body or paano siya na-inherit. So this is referred to as the random X inactivation, which is a little bit advanced. Probably we can discuss that in our topic on December 9. Kasi medyo advanced na siya na topic. So whoever asked this question, thank you so much for bringing this up. So gene interactions, your genes are like humans. They are not islands. They have to interact with one another to ensure that your physiology and your bodily functions are all okay. So there are multiple types of interaction. So what was discussed yesterday is allelic interaction. This is the interaction within the gene pair that is commonly observed in heterozygotes. Kasi sa heterozygote state natin, there are 2 different alleles in that particular configuration. All right? So meron tayong complete dominance, wherein one copy of the dominant allele is enough to elicit the trait. You have incomplete dominance, wherein the phenotype of the heterozygote is actually at the midpoint of the 2 homozygous extremes. So say, for example, one homozygous is red while the other homozygous is white. The heterozygote will ultimately be pink because there is interaction between the gene products. Ayan siya. Also, na-discuss din natin codominance, which is very different from incomplete dominance. So, sa mga maglelet dyan or sa mga mag-enmat dyan, so at least this question is commonly asked. So, what's the difference between incomplete dominance and codominance? Overdominance this time is when a heterozygote outperforms homozygotes. So generally, this is used in agriculture, plant breeding, and animal breeding because you would always want to produce individuals that are that perform very well in the field or in plant plots, so on and so forth. So lethal genes, ito another type of interaction wherein if you have a particular allele for a particular genotype, this will automatically lead to death, such as the Manx cat as discussed yesterday. All right, so lahat ng prinsipyong yun, diniscuss natin, ay pinapalit sa akin yung notability. Sige, submit. Okay, cancel. Okay. There, so lahat ng prinsipyong yun ay comes with the understanding of how chromosomes behave and what is in the chromosomes. upon looking at mitosis, na-appreciate natin how chromosomes are maintained across different cell lines. And looking at meiosis, nakikita natin paano na-form yung haploid na gametes natin. But at the end of the day, your chromosomes are nothing more but DNA. So, can anybody tell me what are the 4 major biomolecules? What are the 4 major biomolecules? So these 4 biomolecules are, if you recall basic biochemistry, these include proteins. Ayun, may sumagot. Proteins, lipids, carbohydrates, and lastly, your nucleic acids. Thank you so much. So these 4 are the major are the major biomolecules found inside the cell. So kung hihimay-himayin mo yung cell mo, all biomolecules can be classified across these 4 categories. But what's so special with this is that among these 4, only one happens to be the informational molecule. And when we say informational molecule, it is the one capable of carrying information from one generation to the next. Your proteins cannot do that. Your lipids cannot do that. Neither can your carbohydrates. Only nucleic acids have the ability to confer information that can be interpreted by the machinery of the cell itself, making nucleic acids set apart from the rest of these 4 biomolecules. So for today, we will be looking at the informational molecule from a biochemical perspective. So I hope ready yung basic organic chemistry and biochemistry na concepts, kasi we will be tossing concepts around that concern the nucleic acids. So here makikita natin that your chromosomes are actually DNA in nature. So your chromosomes is a combination of your DNA and at the same time, your packaging proteins. Bakit importante na ipapackage natin yung DNA into the chromosomal configuration? Bakit? It is because of the fact that if your genetic information will simply exist as a double-stranded DNA, it is very, very fragile. And kung very fragile siya, remember, your DNA has to be must contain very high integrity so that it can be passed down from one generation to the next. But if marupok siya and very madali siyang ma-chop off, therefore, hindi siya magandang conduit of information. So to make sure that your genetic information is stored and maintained, kailangan siyang i-package. And this is made possible by packaging proteins. With the addition of packaging proteins, your DNA becomes fully condensed in a more protected state that is in your chromosomes. So the chromosomes become further housed inside the nucleus so that it can be protected from the harsh environment in the cytoplasm. Kasi remember, sa cytoplasm, andyan yung cellular machinery and at the same time, very active yung enzymatic activity dyan, such that you have their enzymes that can degrade the genetic material. So there. So that's the genetic material. As we said a while ago, our star for this discussion involves your nucleic acids. So your nucleic acids, just like any other type of biomolecule, is a polymer in nature. Kapag sinasabing polymer, anong ibig sabihin ng polymer? When you say something is a polymer, it is composed of repeating monomeric units or repeating monomers. And these monomers come in the form of your nucleotides. So yung nucleotides natin are the monomeric units that if pasted together right next to each other, this will make up the sequence, a very long sequence that will comprise nucleic acids. So your nucleotides are our monomers. So as monomers, they must contain specific parts and components. So your nucleotides contain 3 specific parts and components. So, sorry about that. Ito. So, your nucleotide is composed of three, di ba? Sabi natin. First, you have the phosphate backbone, followed by a 5-carbon sugar, followed by a nitrogenous base. Yan siya. Your nitrogenous bases. And these three components are bonded together by virtue of covalent bonds. So the bond that is responsible between your phosphate backbone and your ribose sugar, or rather your pentose sugar, is referred to as the phosphoester bond. And the bond that is found between your 5-carbon sugar and your nitrogenous base is your glycosidic bond. There you have it. So phosphoester bond and your glycosidic bond. So makikita natin dito that these nucleotides are held together by covalent bonding. So makikita din natin that each specific parts will ultimately play a role. So first, let's take a look at your phosphate backbone. So your phosphate backbone natin, so lagyan natin dito. So phosphate backbone. So eto yan siya. So your phosphate backbone is comprised of phosphorus covalently bonded with oxygen molecules. And because of this particular behavior of phosphate to create these bonds with oxygen, it makes your DNA negative. It makes your nucleic acids in general negatively charged. So kung titingnan natin yung DNA natin, these are actually negatively charged biomolecules. And on top of that, your phosphate backbones are the ones responsible for connecting one nucleotide to another nucleotide. So connects multiple nucleotides. So in short, di ba nga sequential yun siyang nucleotides natin? Your phosphate backbone is the one responsible for that. Next is your 5-carbon sugar, as shown here. So this is your 5-carbon, also known as your pentose sugar. So pentose is a general term that describes carbon, or rather sugars with 5 carbons. So itry natin i-trace kung saan yung 5 carbons na to. So you have here central oxygen. So this is your first carbon, your second carbon, third, fourth. Pero wait, babalik siya dito sa central oxygen. Ayan siya. So therefore, there is a 5th carbon that protrudes outside. Ayan siya. So for the benefit of biochemists, generally they name the carbons here with an apostrophe. So this is the first prime carbon, second prime carbon, third, fourth, and fifth. Yan siya. So this is generally placed so that parang ma-identify lang yung mga carbons. Kasi kung titingnan natin yung nucleotides, ang dami-dami pang carbons nito if i-consider din natin yung nitrogenous bases. Ayan siya. So this is a 5-carbon sugar. Tapos generally, one major importance of the 5-carbon sugar is that this serves as the central. Pero hindi naman siya super central. Pero this connects the entire components of the nucleotide. With one another. Kasi without the 5-carbon sugar, there is no way for us to connect the phosphate backbone to the nitrogenous bases. Ayan, so very important siya guys kasi parang siya yung pinaka-central hub that is connected to the phosphate and that is connected to the nitrogenous base. So this also plays a large role in maintaining DNA or RNA integrity. Alright, so na-discuss na natin yung phosphate backbone. yung 5-carbon sugar or your pentose sugar. And at the same time, i-discuss din natin yung pinaka-importanting part ng nucleotide, which happens to be your nitrogenous bases. Alright, so your nitrogenous bases are the ones that are generally found, and its sequence contains the information. So in short, kung babasahin mo yung yung genetic code natin, this is nothing more but a sequence of A, T, C, G, so on and so forth. And these A's, T's, C's, and G's are the nitrogenous bases that we know of. So your nitrogenous bases are generally characterized in such a way that your adenine will pair with thymine, so adenine to siya. And your cytosine will pair with guanine. With guanine. There you go. So, this is one major importance of your nucleotides, yung kaniyang specific base pairing, which we will come to appreciate as we discuss further topics in the central dogma of molecular biology. So, eto, makikita natin yung very important components ng monomers ng ating nucleic acids. So regardless if DNA man yan or RNA man yan, which are the two types of nucleic acids, these will always contain the following, which are your phosphate backbones, your 5-carbon sugar, and your nitrogenous bases. So later, we will discuss more on how these will contribute to the overall structure of the DNA. But for now, at least na-discuss na natin kung ano yung itsura ng isang nucleotide. So, we now have discussed the nucleotide. It is important for us to compare the two types of nucleic acids. Siguro naririnig nyo lang to yung two types of nucleic acids, no? But it's time that we explore further kung kailan siya ginagamit ng cell at kung kailan siya hindi ginagamit ng cell. Two types of nucleic acids. So, the two types of nucleic acids are classified as either DNA or RNA. Okay, so DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid only. So let's compare the two. So kung titingnan na natin sa pangalan, meron ng pagkakaiba. Etong deoxyribonucleic and ribonucleic acids. So kung titingnan natin yung DNA, it is coined as deoxyribonucleic acid because number one, of certain differences in the ribose sugar, or rather in the pentose sugar. So according to the category of the pentose sugar, makikita natin na yung difference ng DNA is that, sige, i-draw muna natin yung pentose sugar ng DNA. So if this is the pentose sugar of your DNA, so remember may fifth pa yan siya. And at the same time, duplicate natin siya sa RNA natin. Here. Yan siya. So if we are going to take a look at the sugar alone, each carbon contains a hydroxyl functional group. Ayan siya. So, meron niya siyang OH. There. So, one major differences of DNA and RNA is that in the deoxyribonucleic acids or DNA, tawag sa kanya deoxy kasi wala siyang oxygen in the second carbon. Ito. Instead, it will just contain a hydrogen atom. Yan yung isa sa mga major differences natin. But kung titingnan natin yung RNA, yung kanyang sugar lang on its own will still contain an OH group at the second prime carbon. Alright, so this is one major difference. So the sugar is deoxyribose in DNA, deoxy because it is devoid of an oxygen at the second prime carbon, and at the same time, yung pentose sugar natin sa RNA is simply a ribose sugar. Ribose versus deoxyribose. Ribonucleic acid versus deoxyribonucleic acid. So that's one major difference. All right. So next is, makikita din natin yung differences between a DNA and RNA kung saan siya nahahanap at kailan siya ginagamit ng cell. So your DNA is more permanent as compared to RNA that is relatively temporary. So yung DNA mo, palaging andyan yan siya as long as your cell will always have the nucleus and your chromosomes. And these are found in your chromosomes. Whereas your RNA are temporary and will be found outside the chromosome, such as the cytoplasm or in other parts of the cell. But these are bound to be degraded once it has already fulfilled its function. Whereas your DNA has to be stably maintained throughout the life cycle of the cell. Ay siya, permanent yung DNA, temporary yung RNA. And this can be attributed to the fact that your DNA is relatively more stable as compared to your RNA that is unstable. Kasi mas madali siyang ma-degrade and mas madali siyang mawala sa cell. So permanent, temporary. Next, number 3. We can also look at the DNA by virtue of its strandedness, kung ilang strands. So the strands in your DNA are generally double-stranded. So kapag double-stranded, ito yung makikita natin na yung DNA natin is comprised of 2 strands that is running anti-parallel against each other. As compared to your RNA, na single-stranded lang yan sila most of the time. Yan siya. So single strand lang yung RNA natin. So this is also one of the major reasons why your DNA is more stable because of the double-stranded configuration. Mas napoprotektahan niya yung nitrogenous bases as compared to the RNA that is relatively single-stranded. Ayan siya. So single strands versus double strands in RNA and DNA. And lastly, in DNA, bases natin include your adenine that pairs with thymine. At the same time, your cytosine that pairs with guanine. Whereas in RNA, you still have adenine, but this time, thymine is replaced with a uracil, and cytosines and guanines are still found in the sequence. So this is one major difference, the presence of a thymine and a uracil in DNA versus RNA. So ito, na-distinguish na natin yung DNA and yung RNA. So later makikita natin in the process of the central dogma of molecular biology kung kailan na-produce yung RNA in the process of transcription. Okay, so I hope these 4 major characteristics shed light kung ano yung differences between a DNA and an RNA. So guys, kamustahin ko kayo? So far so good? Do we have questions coming from the class? Wala naman. All right, I'll take that as none naman so far. Thank you. So let's focus on the structure of the DNA, because your DNA is the material responsible for heredity. Ayan siya. And your DNA is the hereditary material. So it is just right that we focus on the DNA. So your DNA contains multiple characteristics. Number 1, your DNA is relatively anti-parallel and double-stranded. Guys, ano ibig sabihin ng anti-parallel yet double-stranded? So we can imagine that the DNA is double-stranded. Ayan siya, as shown here. So, for example, nitrogenous bases natin ganito. So, A pairs with T and C pairs with G. So, we can see that we have two strands here. The strand represented here at the left and the strand represented here at the right. So, double-stranded siya, yes. Gets yung part na yon. But what about anti-parallel? What does it mean to be anti-parallel? Anti-parallel means that it is running in the same, or rather, means that it is found on the same dimension. So yung dimension runs towards this particular direction. However, the directionality of the two strands are different. So this can run in one direction, and the other complementary strand will run opposite against it. So makikita natin the same axis, different directions. That is what is meant by anti-parallel. So being anti-parallel and double-stranded is the major characteristic of your DNA. So let's try to take a look at it further. So bakit kailangan siyang, rather, anong ibig sabihin ng anti-parallel and double-stranded? So I'll erase this one. So let's take a look at a sequence that will feature the specific components of the nucleotide. So as much as possible, I'd like you guys to draw with me so that kahit kayo ma-master niyo rin siya. So let's draw one linear sequence first. So we start with a phosphate backbone, followed by a ribose sugar, followed by a nitrogenous base. Say this is thymine. So this is followed by another phosphate backbone, ribose sugar. Say example, this is an adenine. Followed by another nucleotide. Say for example, this is guanine. Okay, so remember, these are all connected by virtue of covalent bonds. Ayan siya. Okay, so looking at this strand alone, we can already see a particular direction na may pinapapuntahan siya. And that direction is running from here to here on the basis of the particular, anong tinatawag natin? On the functional groups that is associated with carbons. Ang ibig sabihin ng functional groups associated with the carbons. So, i-drawing pa natin na mas malapitan. So let's label the carbon so that this will be made clear to us. So this is carbon number 1, carbon 2, carbon number 3, carbon number 4, and somewhere here is carbon number 5. Alright? So sa part na to, ano yung pinakadulong carbon na makikita natin? So this is the 5th carbon as highlighted. So this end here is referred to as the 5' end because the last carbon that you will encounter in that particular part is the 5th carbon in the pentose sugar. So this is the 5' end. Okay. Clear tayo dun? So I hope this is clear. So this is also known as the phosphoryl end. Because yung functional group na associated with the last carbon is a phosphate group. Okay, so this is the 5' phosphoryl end. So ano yung kabilang end therefore? So let's label the carbon. So this is carbon number 1, 2, 3, 4, and somewhere here is carbon number 5. So ang last na carbon na makikita natin sa sequence dito is actually carbon number 3. Siya yung pinaka nasa dulo ng sequence natin. So kung 5' end kanina, anong end to? Bale. So this is a 3' end. Alright. And this is also referred to as, so ano yung functional group associated with this? So it's an OH. So therefore, this is a 3' hydroxyl hydroxyl end. So kung titingnan natin, yung sequence na to runs in the 5' to 3' direction. Okay. Is this clear? So guys, dahan-dahanin natin kasi medyo important siya na concept. So, pa-thumbs up if nag-gets yung concept ng 5' to 3'. Klaro siya? So, saan nanggaling yung basis ng pag-number? The basis is on the carbon and also on the functional group sa end na yun. So, 5' end, yung pinaka nasa upper end, and associated with it is the phosphoryl group or the phosphate backbone. And yung pinaka-bottom dito is your 3' end kasi yung pinaka-last na carbon dito is the one containing the hydroxyl end. Okay. So, natapos na natin yung isang part. This runs in the 5' to 3' direction. Anong ibig sabihin bale ng kanyang complementary strand or the strand right next to it? The strand right next to it must therefore run in the opposite direction. So, if the direction here is 5' to 3', so babaliktad yung other strand such that if we draw this, it will look like this. If we will draw the second strand, it will look like this. So, guanine will pair with cytosine, and this time, yung ating sugar will look like this, and your phosphate will look like this. All right, so adenine will pair with thymine, bonded with its own sugar and its own phosphate backbone. And thymine here will pair with adenine. Oh, sorry. And so I hope you guys are drawing along. So ayan, makikita natin. There. So in this particular sequence, let's try to identify the direction. So sa upper end nito, ano yung last na carbon na makikita natin? So let's draw this part over here. So this is the first carbon, second, third carbon. So therefore, third carbon yung pinaka-last, making this the 3' hydroxyl or the 3' end, or simply known as the 3' end. So here, 3' end siya. Whereas, so this therefore is the 3' end. So tingnan natin yung pinaka-next na kabila. So eto, 1, 2, 3, 4. Somewhere here is 5. So anong end to? So this therefore is the 5' end. Ayan siya guys. So at least ngayon naintindihan na natin bakit yung DNA kapag sinasabi merong 5' to 3' end. So if this is the DNA sequence, na iniba lang natin yung orientation. So this is the 5' to 3' end, and opposite it is the 5' to 3' end running in the opposite direction. So clear na siya guys, bakit anti-parallel yung DNA? Kasi the directions of the strands will vary from one complementary strand to the next. Technique dito is titingnan nyo lang talaga kung ano yung pinaka-last na functional group sa pinaka-last na nucleotide na makikita nyo. Okay, is this clear? Klaro tayo? The double-strandedness of the DNA. Okay. So, this is one major characteristic of your DNA. So, anti-parallel siya and double-stranded din siya. And at the same time, let's take a look at the concept of DNA packaging. So, ano nangyayari sa DNA packaging, guys? Sa DNA packaging, your DNA will condense from a double-stranded sequence all the way to a very large chromosome. So, this involves a very meticulous process that will involve packaging proteins. So let's take a look at the process of DNA packaging. So sa DNA packaging, first, this is your naked DNA. Ayan siya. So this is your naked DNA, and on its own, DNA is very fragile and prone to degradation. So this must be associated with the next class of proteins, which is referred to as your histones. And your histones are globular proteins that are composed of multiple units. And what happens here is that your DNA will wrap around these histones. And in its wrapping, makikita natin that your DNA will start to condense. So from a very flimsy, naked DNA structure, magsa-start na siyang magkaroon ng magkaroon ng relatively higher level of protection. So as it condenses, yung mangyayari niyan is that this will ultimately form the beads on a string configuration, wherein your DNA is associated with these histones. Ayan siya. So this is referred to as the beads, beads on a string configuration, as shown here. So beads on a string configuration yan siya. So repeated units of the DNA with the histone, and each unit here is referred to as the nucleosome. Yan siya. So nucleosome yung tawag natin dyan. So after this, hindi pa siya nagtatapos yung ating DNA packaging. After the beads on a string configuration, yung ating histone proteins, the individual histone proteins here, will cluster together and clump in such a way that this will produce the solenoid structure. Because the histones are also attracted to one another, magkakalapit at magdidikit-dikit yan sila, such that this forms the solenoid structure. And this resembles the wires we see on telephone cords. Yan siya, the solenoid structure. So here, the nucleosomes themselves will cluster on top of each other in a spiral-like manner, resembling that of a telephone wire. So the solenoid structure. So makikita natin na nag-condense pa siya further, no? So this is another level of DNA packaging. And apart from this, madadagdagan pa siya ng isang layer of packaging. So we can imagine that at this point, condensed na yung DNA natin. So this can be further condensed by forming loop domains. Etong solenoid structure natin. So this is now your loop domains. And what is observed here is that your solenoid structure will form these loops, and these loops are based on this sheet of protein we refer to as non-histone proteins. Non-histone proteins. So yan siya yung solenoid structure all the way to your loop domains. So makikita natin from naked DNA, ito siya, to the nucleosome, which is the beads on a string configuration, to the solenoid, so pakapal na siya ng pakapal guys, to the loop domains. And ultimately, these loop domains will form your chromosomes as we see them. So yan siya. So medyo mahaba yung kwento from the naked DNA all the way to your chromosomes found during mitosis or meiosis. Okay? So this is DNA packaging. So kung titingnan natin, the journey of the DNA is not very simple. However, its effects in such a way that it packaged itself to such state of stability makes it possible for the genetic material to be passed on from one generation to the next and to be maintained across our lifetimes. So because of this packaging, because of the DNA packaging phenomenon, our DNA are protected from any forms, or are mostly protected from most forms of mutagens. So, ayun siya. Makikita din natin that this contributes to genetic stability in our lives. So, ayun. So, this is for DNA packaging. Okay, so question so far sa ating nature ng DNA and how it is transformed into your chromosome structure. Kamustahin ko muna kayo, guys. So far, so good? Kamusta? Or any questions coming from the chat box? Sir Josh, may mga questions ba sa chat box?
None po, sir. Check ko po sa
Sa
Facebook.
FB, may address na po natin.
Mukhang wala naman po, sir.
None so far. Okay, sige. Okay, so we now proceed with the central dogma of molecular biology. So na-discuss na natin yung nature ng ating genetic material. It is about time that we also discuss kung ano yung nangyayari, how it is interpreted. Remember that your DNA is the informational molecule. One function of information is, number one, it should be able to maintain itself, and at the same time, it should be able to be interpreted. Otherwise, the information will only remain as information without being put to actuality. So kailangan natin siyang i-maintain at kailangan natin siyang basahin. Otherwise, hanggang information lang siya. Hindi na siya magagamit ng cell. So this is where the central dogma of molecular biology comes in. Bakit siya, guys, tinatawag na central dogma of molecular biology? This is called the central dogma of molecular biology because dogmatic siya. It is like all life forms will follow these particular principles so that life will perpetuate. So that is how life on Earth has evolved, by following these principles. And these principles include, number one, starting with your DNA, it should be replicated so that it can be maintained, so that the genetic material can be stably maintained and passed on from one generation to the next. And this is made possible by the process of replication. Ayan siya. So replication siya. This is for maintenance. Light natin ng color red, yung maintenance. Okay. So your DNA has to be read and interpreted by virtue of transcription. Kasi yung nangyayari dyan, guys, is that your genetic material is found in the nucleus, which happens to be your cellular library. So for the cell to be able to take out whatever information that is present inside the library, it has to take out a copy of that information. And the process of creating a transient copy of whatever information that is present in the DNA is referred to as transcription. That transient copy is in the form of your RNA. Okay? So your RNA will then serve as a template for the process of producing the gene products that is being coded by your DNA. And this is referred to as translation. So your translation will produce a polypeptide chain that, upon further processing, will produce your functional proteins. So yan siya, guys. So bali, from DNA to RNA to polypeptide chain to functional proteins. And these proteins are the cellular machinery that makes enzymatic processes possible, and also maintain the structure of the cell, and also the ones responsible for the phenotype as observed, say for example, in Gregor Mendel's experiments. So from DNA to RNA to polypeptide chain to functional proteins. We involve the process of replication to maintain the genome, and transcription and translation to interpret the genome. Okay, so this is the central dogma of molecular biology. So we start with the first process that is referred to as replication. So replication is a process wherein we create an exact copy of the entire genetic material. Okay, we create an exact copy of the entire genetic material. So by replicating your DNA, dito na, remember this occurs at what stage in the cell cycle? This occurs at S phase of the cell cycle, if you recall. And in this process, nadodoble yung DNA content natin. So replication requires a lot of proteins, and our major protein here is the DNA polymerase. So your DNA polymerase is the major protein. So let's start by illustrating what happens during the process of replication. So this is a relatively complex process, but for the sake of basic genetics, simplify lang natin siya as much as I can. So we start with the DNA. As shown here. So the DNA, remember, has a 5' to 3' direction and a 3' to 5' direction. Okay. So the start of replication will always begin by ano nakikita niyong problema dito? It is still in a double-stranded state. So yung double-stranded state niya, kailangan siyang i-unwind and change into its single-stranded state. So by changing it into a single-stranded state, kailangan natin i-disrupt yung hydrogen bonds between the nucleotide bases so that you will ultimately transform this into a single-stranded DNA. So titingnan natin to. So upon the introduction of a special enzyme we refer to as helicase. So 3'. And this is 5'. So check natin kung na-maintain. 5' to 3', maintained. 3' to 5'. So na-maintain yung anti-parallel ng DNA natin. Okay, there. So makikita na natin dito that from the double-stranded DNA na state, naging single-stranded na siya. So these are now the templates for DNA replication. So the enzyme possible for this particular process is referred to as the helicase. So yung helicase natin unwinds the DNA and ensures that there will be single-stranded templates for the process of replication. So that is where helicase comes in. But the problem with the DNA is that it has the tendency to wind back. Pwede siyang bumalik ulit sa kanyang double-stranded state. So papasok dito yung other proteins, and these other proteins will hold the DNA in such a way na i-maintain niya yung single-strandedness niya. And this is referred to as the SSBPs, which stands for the single-stranded binding proteins. So may helicase, may single-stranded binding proteins, and at the same time, meron tayong another enzyme here upstream of the helicase that allows the DNA to relax at this particular point. So as your helicase goes through the sequence, eto namang gyrase, your DNA gyrase is the one that ensures that kung ano man yung madadaanan nitong helicase will be protected from additional stress brought by the unwinding of the DNA. So that is your gyrase. So may helicase unwinds the DNA. Gyrase relaxes the supercoils upstream your helicase. And SSBPs, your single-stranded binding proteins that ensure that your DNA will hold on to its single-stranded state. So SSBPs. All right. So ready na yung DNA natin for replication. The next enzyme na papasok are your primases. So your primase are very special enzymes in such a way na magdadag siya ng short RNA primers. Yung short RNA primers natin will bind to the sequences here. So ito makikita natin. Okay. So short RNA sequences lang yan siya na very important. So these are short RNA primers. So these are RNA sequences that are added onto the DNA. So these will be temporary primers and will be removed later in the process. So ayan. So remember that your DNA runs in the 5' to 3' direction for the upper part. So kung 5' to 3' to guys, ano yung magiging direction ng RNA primers natin? Remember, it has to be anti-parallel. So this is the 5', which is opposite the direction of its template. So 5' to 3'. Whereas this one, opposite the direction of its stem. Okay, so I hope this is clear. So this is your primers added by your primases in an anti-parallel orientation with the template DNA. Okay. So kung titingnan natin to, we can proceed with the action of your DNA polymerase. Pwede na natin ipasok yung ating DNA polymerase. So yung DNA polymerase natin is very special kasi pwede na natin pwede na siyang pumasok sa ating buong complex. So once the DNA polymerase has been recruited because it has recognized na meron ng RNA primers sa ating sequence, pwede na siyang magdagdag ng additional DNA strands. One problem with DNA polymerase is that it generally adds at the 3' end of the sequence. So ayan siya. So magdadagdag siya ng nucleotides at the 3' end of the sequence. So in this strand over here, dito sa taas, in this specific strand here sa taas, eto, so saan siya magdadagdag bali? Sa 3' end ng RNA primers natin. So dito siya magdadagdag in this direction. As such. So that is your DNA polymerase adding at the 3' end of the primer. So here makikita din natin, so kung nasa kabilang strand na tayo kunwari, so saan siya bali magdadagdag ulit? Magdadagdag siya at the 3' end of the primer. So dito magdadagdag yung ating DNA polymerases. Okay. So this strand here will continuously add nucleotides as your DNA unwinds in this direction, brought by the action of your helicase. So habang nag-unwind siya, nakatrail lang to sa likod. Etong strand na to as it is polymerized by the DNA polymerase. But the problem here is that as this sequence unwinds, sa upper part naman, lalaki yung gap na to. Kasi hindi siya mapupunan ng ating DNA polymerase kasi yung DNA polymerase natin is palayo sa ating, yung kanyang orientation. So notice how this strand over here, the movement of the DNA polymerase is continuous and very importantly, towards the replication fork. So because of this, we refer to this strand as, can anybody guess? Familiar with the term leading strand? Yan yung reason bakit meron tayong leading strand. As shown here. Whereas, yung kabilang strand naman guys, makikita natin, eto, that the DNA polymerase is not going to work continuously because it is away from the replication fork. Kasi remember, the orientation of the 3' end kung saan siya dadagdag is away from the replication fork. So this makes this particular sequence here the lagging strand, if familiar kayo. Ayan siya. So leading strand and lagging strand. Leading strand if the synthesis of the DNA polymerase is continuous and towards the direction of the replication fork. Lagging strand if the synthesis of the DNA polymerase is not continuous and away from the replication fork. So let's imagine the same scenario. So kunwari, humaba na yung ating as shown here. So say for example, we have the same strands 5' to 3' and 3' to 5'. Tapos andito sa area na to yung replication fork. Okay. So 'di ba humaba na yung ating sequence. So andito kanina yung RNA primers in the 5' to 3' direction, and since sa 3' magdadagdag yung DNA polymerase natin, so synthesis is continuous, dire-diretso lang, as your helicase will continuously unwind the replication fork, or rather the double-stranded DNA. Tingin tayo sa lagging strand sa taas. Yung lagging strand sa taas, so saan yung RNA primers natin? Ayan siya. So, RNA primers natin runs in the opposite direction. Kasi 5' to 3' yung sa taas, therefore 3' to 5' yung nasa baba. So, saan magdadagdag? Yung DNA polymerases natin. In the 3' direction. So, dadagdag siya dito, and dito, and dito. Towards this direction. So, since sa leading strand natin, walang problema kasi magtutuloy-tuloy lang yan siya. How about this part over here? Sa ating lagging strand. This is what we refer to as an Okazaki fragment. Ayan siya. So the Okazaki fragment will have to be primed again. So papasok ulit yung ating RNA primers by virtue of the enzyme primase so that pwedeng pumasok ulit yung ating DNA polymerase 3. Ayan siya guys. Ayan, so makikita natin dito na very important yung ating pagpa-prime ulit, pagdadagdag ulit ng ating short RNA primers in the lagging strand. So there. So eventually, at the end of the process, you will have this particular structure in the lagging strand, and there is very minimal problem with the leading strand. So this is, label natin ulit para di tayo mawala. This is the lagging strand and the leading strand. So ano yung problem na ating makikita pa dito? Another problem here is that the DNA, as shown, is not purely DNA. Because this contains sequences that are RNA in nature because of the RNA primers that were added by the primases. So for us to maintain DNA to DNA interaction na purely DNA lang talaga yung nasa genome natin, this need to be replaced. So this will be replaced by another set of enzymes such as your DNA by the way guys, yung DNA polymerase is specific to DNA polymerase 3. So DNA polymerase 3 yung ating major enzyme, yung nagdadagdag kanina ng nucleotide. Yung next na enzyme na papasok is your DNA polymerase 1. Yung DNA polymerase 1 natin will remove the RNA primers from the sequence and replace them with new DNA, as shown here. There. So this will replace RNA primers with new DNA. Okay, so DNA polymerase 1. And also, papasok yung isa pang special enzyme to repair these particular nicks we often encounter, yung mga nasisirang phosphoester bond natin, and add the remaining phosphoester bond that needs to be added to the sequence. So what happens at the end of replication is that first we started with a double-stranded helical structure. Eventually, na-unwind siya. And in its unwinding, na-expose yung nitrogenous bases niya. And by this, we add new strands to the sequence. This is the main reason why DNA replication is said to be semi-conservative. Ano ibig sabihin ng semi-conservative? It is semi-conservative, semi lang, half lang siya, because only half of the newly synthesized DNA strands after replication are of parental origin, whereas the other half are newly synthesized. Kaya this is one major characteristic of DNA replication, it being semi-conservative. Okay, so that's for the process of replication. Do I have questions from my audience? May tanong ba, guys? So clear, no? Paano nagkakaroon ng leading strand and lagging strand? Very important siya. Or may gusto ba kayong i-clarify or may gusto ba kayong tanong? From the FB Live, Sir Josh, meron ba?
None po, sir.
None naman so far. Okay, so we proceed with the next process in the central dogma of molecular biology, which is transcription. So I'll share my screen again. Okay, so this is now the second process, no? So transcription. So guys, you can imagine your DNA to hold lots and lots of information. And this information, so say for example, if this is your DNA, so this is highly informative. So this contains instructions for the phenotype. This also contains instructions to how enzymes are made. This also contains instructions on how to fight off infection and how we age. So the DNA itself is highly informative. However, this information is just stored in the nucleus. So ito yung cell, lahat ng information na to and lahat ng pagha-hype sa kanya are all present inside the nucleus only. So remember that most of the processes in the cell occur in the cytoplasm because of the multiple structures that are present there. But the information is just found inside the nucleus. So it's for the cell to take out the information from its nucleus kung kailangan niya, like the DNA itself. The answer to that is no, because it is very impractical for the cell to take out a very, very large, let's say, a very, very large conduit of information na ilalabas niya sa cytoplasm just for it to read a few pages. So therefore, the process of transcription enables the access to this information in your DNA by using specific enzymes that can produce RNA copies of the instructions from the DNA. Ayan siya. So, you can imagine na kunwari, if I am a chef, tapos I would like to learn a particular recipe. And that recipe is accessible in the library. So what I'd like to do is to read that information in the library and bring it to my home. So ang gagawin ko, pupunta ako sa library to take out that particular information. So once in the library, sinabihan ako ng guard na, "Sir, bawal mo yan ilabas." So what's the smartest thing for me to do? I will create a photocopy of that particular section which contains the recipe that I badly needed so that I can leave the book inside the library and I can take with me a photocopy of the information. So the book is the DNA and the photocopy is the RNA. So once I already have a photocopy of that information, pwede na akong lumabas and pwede na akong umuwi sa bahay ko and I can cook that particular recipe. All right. So that is the main concept of transcription. You have to produce transient copies of the permanent information that is found in the DNA. And these copies are in the form of RNA. Okay. So let's take a look at how a gene looks like. So remember in our previous discussion, that if this is the chromosome, this will contain particular loci that contains information. So may mga loci yan dyan siya. Or yan. Okay, so take note that a large percentage of the chromosomal arms do not contain, let's say, do not contain information for a particular gene product. This is because of the fact that not all sequences in the DNA will be coding for a gene product. Say, for example, if these regions coded in pink will code for flower color, for a pigment that will specify a specific flower color, these regions here na walang any type of information are not coding. So therefore, the process of transcription will not require the entire sequence of the genome, just the location kung saan nako-contain yung genetic information. This is one major difference with replication. Remember, sa DNA replication, lahat-lahat na yan. Kailangan mo i-replicate. Whereas in transcription, you will only transcribe certain regions in the genome that will code for a particular gene product. Studies have it that around 25% of the entire genome codes for a product, while the remaining 75% is simply there for structural purposes and do not necessarily code for a gene product. So here makikita talaga natin na hindi lahat-lahat ng sequences in the DNA will be transcribed. So, a transcription unit is a region that will be transcribed. So, say for example, this is one very long stretch of DNA. And this long stretch of DNA, sa part na to, andito yung gene for, let's say, gene for yellow pigmentation. So this part right here will be transcribed and will form one transcription unit spanning the entire sequence. Whereas this location here, which does not code for anything, is not a transcription unit. So let's take a look at the transcription unit up close and personal. So the transcription unit is composed of the following parts. First, you have the regulatory sequences, which are referred to as your promoters. So these promoters are the ones responsible to serve as binding sites for enzymes responsible for the process of transcription. So these are very short sequences in the DNA. Afterwards, preceding the promoter sequence is your coding region. So your coding region will be the one used as a template for transcription. So in short, yung promoter region mo guys is simply there to recruit the enzymes needed for the process of transcription. Your coding region will be there to serve as the template for transcription. And lastly, your terminator sequences found upstream will signal the stop of transcription. And so makita natin, so we have the promoter, the coding region, and the terminator. So let's take a look at the process of transcription, kung paano siya nagsisimula. So the process will start with the recruitment of transcription factors. Ayan siya. So your transcription factors, so remember our transcription unit a while ago, yung isang part doon is the promoter region. So your transcription factors will recognize the promoter sequence. And once it has already found that the gene needs to produce a transcript, it will bind itself with RNA polymerase. So, yung mangyayari niyan is that the transcription factor, which is a protein, will recruit the RNA polymerase at the site of transcription. So, say for example, ito yung RNA polymerase na natin. So, this is now your RNA polymerase. Okay, so this is your RNA polymerase already at the location of the promoter sequence. The reason why it has bound to the promoter sequence is because of the action of your transcription factors. So kapag nasa sequence na bali yung ating RNA polymerase, so if this is the DNA, so kunwari ito yung DNA natin, it will transcribe the, kung imagine natin, so 5' to 3' to, and 3' to 5' to. It will transcribe only one region of the sequence, which is the 3' to 5' sequence. So, eto lang yung gagamitin niyang template. Yung 3' to 5' sequence, because as it moves along the DNA structure, it has to add RNA in the 5' to 3' direction. Always remember guys, na yung pagdagdag ng nucleotides will always be in the 5' to 3' direction. So as it moves along, it will add complementary nucleotides to your template strand, which is the 3' to 5' strand, also known as the antisense strand. So antisense strand yan siya. Okay, so that's for the process of transcription. So eventually, as your RNA polymerase moves along the sequence, this will produce this particular sequence over here. Ito siya, which is RNA in nature. And as it moves along, parang, ano yan siya guys, maglo-loosen up yan siya from the sequence. And this is your DNA over here. Yan siya. All right. So, i-complete natin yung drawing, guys, by drawing the rest of the nitrogenous bases. There you go. So, makikita natin that your RNA will slowly fall off from the double-stranded helix because DNA to RNA interactions are relatively weak. Okay. So, yung mangyayari dito is that once your DNA has encountered the terminator sequences, so kunwari andito na yung terminator sequences natin. So, terminator. Your RNA polymerase will fall off from the entire structure, and this will ultimately lead to your RNA molecule. So you now have your RNA molecule that is a transient copy of whatever information this gene codes for. So para nagkaroon ka lang ng transient copy in the form of your RNA. Okay? So I hope this is clear. So nakaproduce na tayo ng RNA. So this is the process of transcription. When does transcription occur? Or kailan nasasabihan yung cell na magta-transcribe na siya? The cell will transcribe specific genes whenever the cell needs it to transcribe. Say for example, kung i-compare mo siya sa replication, na yung replication will occur specifically at S phase of the cell cycle. Transcription can occur at any time of the cell cycle during interphase if the cell needs for the gene product. Say for example, ako kaninang umaga lumabas ako to buy breakfast. And upon walking under the heat of the sun, UV rays were striking against my skin cells. So as my cells react to the presence of the UV rays, it will transcribe the genes. It will express the genes via transcription. The necessary proteins that will protect me from the UV rays. Ayan siya. So the gene will be transcribed whenever the cell needs to transcribe the genes. Okay. So that's for the concept of transcription. So let's have a brief review. Ano nangyayari sa transcription, guys? So in the process of transcription, we appreciate the fact that not all sequences in the genome will contain coding sequences or genes that will or sequences that will code for a particular gene product. Therefore, the process of transcription will only occur in genes. So, we also discussed how a particular transcription unit looks like, which contains the promoter sequences, the coding region, and the terminator region, wherein the promoter sequence is the one responsible for the recruitment of your proteins, for the recruitment of your promoter of your transcription factors, followed by the coding region, which will serve as a template, specifically the antisense strand, followed by the terminator sequence, which will serve as a signal to stop the process of transcription. So upon transcription, what happens is that your transcription factors will recruit the RNA polymerase at the site of the promoter region. And once this has already been put to place, the RNA polymerase will move towards the direction of the sequence, and it will use the antisense strand as a template in the process. So as it moves along, it will incorporate complementary RNA nucleotides in the process, thereby producing long strands of RNA, which will fall off in the process of termination once your RNA polymerase has encountered a terminator sequence. So there you will have an RNA product at the end of transcription. So questions so far, guys? So far, so good? No questions? Clear pa naman yung topic so far. Okay, may message na ata tayo. Very clear. Thanks. Okay, so let's proceed with the last step of the central dogma of molecular biology, wherein we will now produce the products from the RNA transcript. Remember, sa analogy natin kanina, nakaproduce pa lang tayo ng photocopy of the information. This time, kailangan natin iuwi yung photocopy of the information sa mga bahay-bahay natin so that we can produce whatever the recipe calls for. So the recipe is the RNA, and at the same time, yung bahay nyo will be the site where the next step of the central dogma of molecular biology will occur. And this is your ribosomes. So the last step is translation. Translation. So what happens with translation is that you will now produce the final polypeptide product, which will eventually become proteins after a series of post-translational modification. And these proteins are the ones that are functional molecules responsible for a trait. Ayan siya. So responsible for a trait. So let's take a look at the process of translation. The process of translation will require all three types of RNA. So the three types of RNA used in the process of translation is the mRNA. So ito yung long sequences natin. So this is the mRNA, which stands for the messenger RNA. And this will be used as the template for translation. And then you also have the tRNA. So yung tRNA mo are very short RNA sequences that have the tendency to fold around each other, which stands for the transfer RNA. And this will contain the amino acids at the site of translation. And lastly is your ribosomal RNA. So rRNA, which complexes together with ribosomal proteins to form your ribosomes. And your ribosomes happen to be the site where where translation will occur. So how does translation happens? So how does translation happen? So translation happens by first we have to start with the concept of the mRNA. So your mRNA is a long strand of RNA sequence that will serve as a template for the process of translation. Okay, so remember RNA na to siya, so 5' to 3'. Tapos, in its sequence, so instead of a thymine, meron na siyang uracil. So, kunwari, ito yung sequence natin. Sorry. Okay, kunwari, ito yung RNA sequence natin. Okay, so say for example, this is our RNA sequence. So your RNA sequence contains nucleotides. Ayan siya, adenine, uracil, cytosine, guanine. Okay, so kung titingnan natin to, this will serve as the template. So in the process of starting translation, the first thing that needs to be accomplished is to assemble your translation unit. And we can do this by assembling your ribosomes to the site of your translation. So upon assembly of your ribosomes at the site of translation, yung mangyayari niyan guys is that the small and the large subunits of the ribosomes will assemble at the site of your mRNA. So remember, your mRNA serves as the template. Okay, so andito na yung mRNA natin. So the next thing is to incorporate your transfer RNA. So your transfer RNA will be the ones responsible to bring your amino acids at the site of translation. So say for example, the sequence we have here is AUG. So therefore, it will call a tRNA with an anticodon loop found in this particular region that will be complementary to your codons in your mRNA. So we will recruit, an mRNA, or rather a tRNA with the following sequences. So kung A to, bali dapat UAC. So UAC is found to carry the amino acid F-met, formylmethionine. Okay, yan siya. So makikita na talaga natin dito na nag-assemble na little by little yung ating protein sequence. So in assembling our protein sequence, your translation unit in the form of your ribosome must move from one place to the next. So magmo-move yan siya, one codon position, here. So it will move along the sequence. So once it moves, it will recruit the next tRNA. So the next tRNA will be very specific to the codon that is recruited by the to the anticodon that is recruited by the three sequences here. So you have, so ano yung sequence natin? So you have, so A, U, U. Okay. So therefore, this will recruit for another set of amino acids. So this is amino acid number 2. Okay? So what's next is that ma-erase to siya, guys, yung tRNA dito kasi matatanggal siya from the equation, leaving only the protein that is necessary to our process, or rather the amino acid that is necessary to our process. So upon the addition of the next tRNA that carries the second amino acid, a peptide bond is formed between the first and the second amino acid. So this peptide bond is made possible by the enzyme peptidyl transferase. And so the peptidyl transferase is the one that establishes the peptide bond between two adjacent amino acids in the sequence. So this will move one codon position again towards the next. And makikita natin dito, mag-move lang yan siya. All right, one sec. There, it moves one codon position. So the next codon will call for a tRNA with an anticodon sequence that is complementary to this sequence over here. So the sequence must therefore be, so therefore C, C, G. So ito siya, guys. Ito yung next na tRNA natin that carries with it amino acid number 3. So ano nga ulit yung next na mangyayari? Matatanggal tong tRNA na to. And at the same time, papasok ulit si peptidyl transferase to establish our third amino acid with the rest of the sequence. So you can see where this is going, guys. Paulit-ulit yun siyang mangyayari. Movement. Movement. And then, it will recruit the next tRNA in the sequence. And upon the recruitment of the next tRNA sequence, you incorporate amino acid number 4. And afterwards, you put in peptidyl transferase to establish the connection between the 4th amino acid with the rest of the sequence. So magpapatuloy-tuloy yan siya, guys, all the way until it reaches a particular stop codon or a particular stop signal. So, kunwari, magpatuloy yung buong sequence natin. So, okay. So, this has continued all the way to reaching this particular point na kunwari, all the way to reaching this particular point na stop codon na yung na-encounter or stop signal na. So, kunwari. Ito siya, guys. So, humaba na yung amino acids natin dito. Amino acid 6. And then, what if the last sequence here is a stop codon? So your stop codons are very specific to UGA, UAA, and UAG. So if the ribosomes will encounter any of these sequences, for example, UGA, it will recruit proteins that will stop the process of translation. So the process of translation is stopped by release factors, RFs, which upon encountering these particular sequences that code for stopping the process of translation will insert themselves in the ribosomes and will destabilize the entire translation unit. So upon destabilizing the translation unit, you now have your proteins or your amino acid sequence, and then you can reuse your mRNA for another round of translation. So you are capable of producing multiple identical amino acid sequences from one mRNA template, provided that it is not degraded by virtue of enzymatic activity. So makikita natin dito na na-produce na natin yung ating polypeptide chain. So this polypeptide chain will be converted into a functional protein after a series of post-translational modifications. And once the protein is now fully functional and mature, pwede na siyang mag-manifest as a phenotype. So say for example, what if the gene we have transcribed a while ago is a gene that codes for the yellow color? So upon transcription of that gene, you produce the mRNA sequence, and the mRNA sequence, will serve as the template for the process of translation, when you will produce this polypeptide that eventually becomes a functional protein that, say for example, is responsible for the yellow phenotype. So makikita talaga natin dito, guys, na the process of the central dogma of molecular biology starts with the instruction and ends with the protein that will exhibit whatever the instruction calls for. Okay? So we have discussed the basic processes of the central dogma of molecular biology, which started with replication, wherein at the end of the process, you will produce identical DNA sequences for the maintenance of the genome. And also we have discussed transcription, wherein you will produce transient copies from the permanent instructions found in the DNA. And these transient copies will serve as your templates for the process of translation, wherein you will produce functional proteins that was coded by the genes in your DNA. Okay? So we have discussed the three processes. Sir Josh, a short break muna tayo. I'll have to refill my
Yes,
glass
sir.
of water.
Sure, sir.
So,
5 minutes po tayo.
we can proceed with mutations in the next session, in the next half. of the meeting. Sir Josh, na-realize ko, 11:30 na pala maya-maya, so I have to cut my break short.
Sir, sige lang po.
See, so we proceed with the last part of our discussion for today, which is relatively complicated, but as much as possible, gawin natin siyang simple. So this is now on the concept of mutations. Ay, sorry, di pa ako nag-share screen. Ito na po. Okay. So when you say mutations, probably you have encountered a lot of representations in social media, mainstream media, movies, and all. Kung ano yung itsura ng mutations. If you watch X-Men movies, and then the opening scene or the opening credits will always involve showing this DNA sequence. Tapos parang may black material na nagko-cover ng DNA which represents mutations. It is very incorrect in such a way that changes in the DNA do not occur like that, na walang black material and all that. It's relatively simple and straightforward. When you say mutations, these are changes in the genetic material. And your mutations can be classified as somatic or gametic. Oh, sorry. Somatic or gametic yung mutations, this simply looks at the location kung saan nangyayari yung mutations natin. Somatic mutations are the types that occur in your somatic cells, whereas your gametic mutations are the types that occur in your gametes. So these are in your body cells, and these are in your gametes. So when you say somatic mutation, say for example, kanina 'di ba naglakad ako sa ilalim ng araw. Tapos kapag na-sunburn ako, it is actually a manifestation that the UV rays of the sun did something bad and mutated my sequences in the skin cells, in my skin cells, such that it has to respond to it by killing away those mutant cell lines. So these types of mutations in the body cells are not heritable. Hindi siya pamana to the next generation kasi wala naman siya sa gametes eh. Whereas mutations in the gametes are the types that are heritable in nature. So kapag yung mutation ay nangyari na sa sperm cell or sa egg cell or sa progenitor ng sperm cells and egg cells, which are your primary, secondary spermatocytes, oocytes, yung mangyayari niyan is that the type of mutation becomes heritable na. So these are the types that persists across generation. But guys, the term mutation doesn't have to be negative all the time, because in all forces of evolution, mutation in the genome. Say for example, the color green na iris ng mata is actually one type of mutation that is very, very rare, such that only a small percentage in the population holds that specific eye color, yung color green na eye color. So, however, there are types of mutations that are deleterious as well. Say, for example, a mutation in the sequence that codes for the hemoglobin protein can lead to the formation of physiologic and structural differences in the hemoglobin that makes your cells, your red blood cells appear sickle in shape, which is your sickle cell anemia. So your mutations can either be good or either be bad, depending on how it will affect your survivability in the environment. So there, that's mutations. So if you will summarize mutations, your mutations can be summarized according to the degree of the mutation. So it can either be nucleotide mutations, wherein the mutations only occur in your nucleotide sequence. So say for example, this is your nucleotide sequence here. Tapos for example, A, T, C, G, C, yan siya. So nucleotide mutations can involve the following. Number 1, insertions or deletions. So insertions or deletions, pwedeng mabawasan to siya ng isang nucleotide or madagdagan siya ng additional nucleotide dito. Let's say between GC, nadagdagan siya ng G. So that leads to insertions or deletions in the nucleotide sequence. So kapag insertions or deletions yung nangyayari, this leads to frameshift mutations. So say for example, if this is your nucleotide, this is your sequence here in the DNA, so kunwari ATG, GCC, AAA. So say for example, and remember that your amino acid sequences are read in triplets or in codons. So say for example, in this case, kunwari na-delete yung G natin. So instead of this particular reading frame, everything has to adjust such that iiba na yung reading frame ng sequence natin. So on and so forth. So this leads to frameshift mutations. So that's the first type of nucleotide mutations. You have insertions and deletions that will lead to frameshift mutations. Also, on top of that, you have nucleotide substitutions. So in nucleotide substitutions, walang dinadagdag, walang binabawas. It's just that your nucleotides can be changed from one type to another. For example, the C here has been reverted to a G. So this is now a type of mutant wherein there is a substitution from a cytosine to guanine in the original sequence. So this generally leads to a lot of possibilities such as missense mutations, nonsense mutations, and sense mutations. So when you say missense mutations, this will code for a totally different amino acid product. When you say it's a nonsense mutation, from an amino acid product, this gets converted into a stop signal. And a sense mutation indicates that the amino acid is still the same. Kasi remember that a particular amino acid can be called for by a lot of different codons. So these are the possible changes in nucleotide mutations. So you notice that the severity of, or rather that the degree of damage in these type of mutations are down to the nucleotide only. Whereas there is another type of mutations, and this is relatively more drastic in terms of its changes. So this will include your chromosomal mutations. So yung chromosomal mutations naman natin involve changing either the structure or the number of chromosomes. So if structure, tawag natin dito ay structural mutations. So in changing the structure of the chromosomes, this can involve number one, sequence duplication or deletion or sequence deletion in such a way na, for example, this is your chromosome with the following genes, kunwari, A, B, C, and D. A sequence duplication can occur in such a way na, for example, A, B, C, tapos kunwari, dumoble yung gene na C dyan, D, because this region over here has been duplicated such that this has led to a mutant chromosome. So you can automatically assume din na kapag sequence deletion, may natanggal tayong particular region in the chromosome. By the way, guys, this is not sequence deletion. This is simply identified as duplication or deletion. Chromosomal, all right. Duplication or deletion. Pwede na pala siya. So these are structural mutations. You also have inversions. So yung inversions naman natin, say for example, this is the original chromosomal structure. Oh, sorry. I-ano natin ito. I-uniform. If this is the original chromosome structure, pwede na mag-invert yung particular sequences such that it will lead to this particular configuration. A, C, B, and D. So makikita natin dito na may nag-invert na regions in na segments in the chromosome. So these are segment inversions. Ayan siya. So inversions, and also lastly you have translocations. So translocations are a type of mutations that generally transfer one segment of a chromosome. Say for example, kunwari, ito yung kanyang homologous pair, tapos meron pa tayo ditong isang homologous pair. So translocations can involve the transfer of particular chromosomal segments from non-homologous sequences. Say for example, yan. So nag-translocate yung particular sequences or particular segments from different homologous pairs. So that is what is meant by translocations. So chromosomal mutations involve structural mutations which can either be segment duplication or segment deletion as shown here, or inversions wherein the linear sequence of your chromosomes have been inverted such that it has changed the orientation of certain genes, and translocations wherein matatransfer natin yung certain parts ng chromosomes to another non-homologous chromosome. So yan siya. So chromosomal mutations in the order of structure. You also have another type of chromosomal mutation that is numerical in nature. Numerical mutations. So this can be further classified as the following. Number one can be aneuploidy. So sa aneuploidy, yung nangyayari dyan is that may dagdag bawas in terms of chromosome number. So remember that our chromosome number is a two-sided equation. So you have the left and the right side. Aneuploidy usually changes the right side of the equation. So highlight natin to as green. So say for example, if a person has trisomy 21, so yung nangyayari dyan is that instead of having just two members in the homologous pair for chromosome number 21, nagkakaroon siya ng pangatlo. So kung titingnan natin yung chromosome number niya, bali magiging 2n equals 46 plus 1, and that 1 here is indicative of chromosome number 21. So instead of having just 46 chromosomes, a person with trisomy 21 or Down syndrome contains an extra chromosome number 21, thereby making the total chromosome number 47 instead of the usual 46. So that is what is referred to by aneuploidy. So you have trisomies wherein nadadagdagan, you also have monosomies. So kapag monosomy naman, nababawasan yung isang homologous pair or yung isang bivalent. So instead of having two members to a homologous pair, nawawala yung isa dyan. So that is referred to as monosomy. So there, an example is the Turner syndrome, wherein instead of having an XX or an XY chromosome for a normal for the normal female and the male sex, yung nangyayari is that nawawala ng isang sex chromosome. So this is the Turner syndrome. Okay, so this is for the case of aneuploidy. And lastly, you have euploidy. Yung euploidy naman, guys, changes the left side of the equation, wherein yung nadadagdag bawas natin dito are the chromosome sets instead of just the chromosome number. So kunwari sa euploidy, you have diploids, you also have monoploids, wherein a diploid will contain two sets for all the chromosomes. A monoploid will just contain one set. A triploid will contain three sets, and a tetraploid will contain four sets. So notice how this time we change the left side of the equation. So this is for euploids. Euploidy is generally used in agriculture and in agronomy and in plant breeding because changing the ploidy level of a plant generally confers, say, for example, sterility, such as your triploid watermelon, which is the explanation behind a seedless watermelon phenotype and a seedless banana phenotype. So ayan siya. So this is for mutations. So there are multiple types of mutations. So you have nucleotide mutations, spanning the nucleotide only, chromosomal mutations, which include the structural mutation, and a numerical mutation, which involves changing the chromosome number. and the chromosome set if it is aneuploidy or euploidy. So this brings us to the topic of mutagens. So your mutagens are agents that cause mutations. So your mutagens are the ones that are capable of changing the DNA sequence itself. So what are the types of mutagens that we have? You have chemical mutagens. And common examples of these are the carcinogens that we know of, say, for example, cigarette smoke. Also, those that are fond of eating foods that have been preserved by nitrites and nitrates are also known to produce yung carcinogens natin, specifically if smoked siya, kasi mas nag-intercalate siya with the DNA. So chemical mutagens are chemical agents that induce mutations. You also have physical mutagens. So these are physical forces that are generally found in nature. Say, for example, there's this study that correlates the consumption of very hot drinks with esophageal cancer. Kasi yung study na to has found out that heat itself will produce apurinic sites in the DNA. Ibig sabihin, if this is your DNA, with the following DNA sequence, yung tendency ng heat or very hot temperatures is that this will generally attack your purines, such as your adenines and your guanines. So, parang buburahin niya yan siya. Yan siya. So, it serves as a mutagen by producing apurinic sites. So, this is one common suggestion sa mga nagtitake ng hot drinks. Generally do not put it at or rather do not consume it at close to boiling temperatures, because you will produce apurinic sites in your sequences along the linings of your throat, which can lead to cancer possibly. So physical mutagens such as heat. Also, as you go out under the sun, UV is a form of physical mutagens that have the tendency to mess up our DNA structure. What else? Physical mutagens. Radiation such as gamma, alpha, and beta radiation. These are known as ionizing radiations, which will ultimately affect the DNA as well. Would you believe that you also have biological mutagens? So these are produced by biological agents. Say, for example, a specific bacteria, such as Helicobacter pylori, is found to produce a stomach lining cancer by producing agents that are capable of changing the DNA structure. So Helicobacter pylori, which is about bacteria. Also, you have viruses that can cause cancers as well. So your mutagens are generally the ones that cause mutations. So these types of mutations are the induced type, because you also have spontaneous mutations. Ito yung mga mutations na basta na lang nangyayari even in the absence of mutagens. And this is generally occurring during the process of replication and during the regular cell cycle. And spontaneous mutations are bound to occur across multiple cell lines, specifically as we age. Kaya if you will compare your cells back when you were young and all the way hanggang sa nag-80 ka, you will notice that in your cells, there's an accumulation of mistakes as compared to the original sequence that you had. And this is because the fact that your cells will ultimately accumulate and accumulate and accumulate mutations, either caused by induced processes or spontaneous processes. So that's for mutations. Okay, so time on my watch is now 11:21. Do I have questions from my audience? May mga tanong ba, guys? Sir Josh, may tanong din ba from the Facebook page?
Wala naman po, sir.
All right. So anyway, sir, I think my time is up sa 11:22 na rin naman
Okay.
pala.
Sige po, Sir Emma. I guess wala na pong questions from our students. Teachers, baka po may clarifications po tayo. Wala na ba po? So, wala na po tayong questions. Going once, twice. Okay, check ko ulit sa Facebook page. Ay, sa Facebook Live, baka meron pala.
Sige po, sir.
Meron pa ba? Let me check po. Facebook. Question. Okay. Wala naman na po, Sir M. Okay.
So far, so good naman po.
So I
Okay.
guess that's all for today po on the molecular basis of inheritance on how our genes function within. Thank you very much, Sir Eman Galang. All right. So now Team LRC would like to take this opportunity to thank our speaker for his valuable time. So allow me to present to our resource speaker this certificate. Let me read the citation. Let me read citation. Next slide, please. Okay. Certificate of Appreciation is awarded to Assistant Professor Emmanuel T. Galang for serving as resource speaker during the GENE-ius Cataloga, a tutorial webinar series on genetics, Session 2, entitled You're My Genotype, How Genes Function Within, held today, December 7, 2022 at the LRC UPLB College, Laguna. Signed by our director, Assistant Professor John Mervin L. Embate. Thank you very much, Sir Eman, for our second leg of our genetics tutorials. Thank you, sir. Right.
Thank you.
Now for thank you, sir. And we still have one more session to go po. All right. Now for the evaluation of this session, please do not forget to answer the evaluation form after this tutorials to receive your e-certificate of attendance. Your comments and suggestions will be deeply appreciated by our team. So the evaluation link is being shown right now on your screen. It's tinyurl.com/GENE-ius with capital GENE, capital eval session 2. All right. And kindly answer it before the 10 p.m. deadline tonight. You can also scan the QR code if you want to be directed right away to the evaluation form. Allow me to place it as well here in our chat box for easier access. All right. So we hope that we all gain inspirations and valuable insights from our speaker this morning. So 2 down, 1 more session to go po sa ating mga participants. So join us on our last session entitled Parallel Minds. So we're going to discuss about the different applications of genetics in other fields of study. So this is an important topic in genetics, how it will be applied to other branches of science, other branches or other components of biology, and many more. So our third session is scheduled on Friday, since tomorrow is a holiday. On Friday, December 9, 2022, same time at 9 a.m. and still together with Sir Eman. All right. So at this point, we would like to thank and recognize our team here at LRC and our GENE-ius Cataloga team, headed by our director, Sir J.M. Embate. On our technical side is our student assistant, Mr. Hans Aguilar. And a big shout out to LRC's members, Ate She, Ate Majeng, Tita Allen, and Kuya Iwok. And a big thank you again to Sir Eman, our very passionate speaker for this morning. Thank you so much, everyone. Thank you, sir. Thank you so much, everyone, for joining us. Stay hopeful, stay healthy, and stay safe. We'll see you again on Friday. Thank you, everyone, and goodbye.